Tired of manual plasmid purification? Introducing the Thermo Scientific KingFisher PlasmidPro Maxi processor. This is the cutting-edge future of plasmid purification. Offering ultimate ease of use, experience a fully hands-free purification process. Redefine efficiency with this fully automated solution. The KingFisher PlasmidPro system features a pre-filled cartridge for effortless advanced transfection-grade plasmid DNA, eliminating centrifugation and pipetting, enabling quick turnaround with minimized errors. Experience the future of plasmid purification with the KingFisher PlasmidPro Maxi processor. Prepare for walk-away Maxi-scale plasmid purification. Learn more at thermofisher.com/plasmidpro. Tired of manual plasmid purification? Introducing the Thermo Scientific KingFisher PlasmidPro Maxi processor. This is the cutting-edge future of plasmid purification. Offering ultimate ease of use, experience a fully hands-free purification process. Redefine efficiency with this fully automated solution. The KingFisher PlasmidPro system features a pre-filled cartridge for effortless advanced transfection-grade plasmid DNA, eliminating centrifugation and pipetting, enabling quick turnaround with minimized errors. Experience the future of plasmid purification with the KingFisher PlasmidPro Maxi processor. Prepare for walk-away Maxi-scale plasmid purification. Learn more at thermofisher.com/plasmidpro.
Hello everyone, and welcome to Enhancing Plasmid Purification Through Automation. I'm Matt Thorne of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please visit thermofisher.com. We do encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time at the end of the presentation, and you can also submit any technical issues there as well if you're having any trouble seeing or hearing the presentation. I'd now like to welcome our speaker, Ron Abruzzese, Scientist 3, Sample Prep Thermo Fisher Scientific, and Marie Gonzalez, Staff Scientist, Molecular Biology. For a complete biography on our speaker, click the biography window on the left of your screen. Ron, you can now begin your presentation.
Thank you for the introduction. Today's agenda, we will be talking about introduction. We'll do an introduction to plasmid DNA and its applications. We'll be talking about a flexible, automated mini-scale plasmid isolations, as well as fully automated maxi-scale isolations. So what is a plasmid? It's an extra chromosomal DNA molecule within a cell that is physically separated from chromosomal DNA and can replicate independently. It has a number of applications in the scientific world, especially in therapeutics. As noted here, we have mRNA synthesis, cell and gene therapy, viral vectors, gene editing, and protein therapy. So the general molecular cloning flow is you find a fragment of interest, you generate it, you clone it into a backbone, plasmid backbone, which is then grown up in a bacterial strain through transformation, which then after growth, you isolate the plasmid preparation from the bacterial culture, do purification, and then analyze the plasmid to make sure it has the sequence that you expected. So this just illustrates that we cover the end-to-end solutions for all plasmid purification and expression, from plasmid design all the way through in vitro protein expression.
So what are considerations for optimal plasmid yield and purity? Picking, and we'll talk about these in a clockwise order, starting with the plasmid design. So picking a plasmid backbone that has a strong origin of replication, also known as a high copy backbone, is preferable to weaker or low copy backbones. Antibody selection, which is incorporated into these backbones, also will affect performance. There are several different bacterial strains that are used in the transformation to grow up the plasmids, some of which have been genetically engineered to enhance plasmid replication within the cells. For purification, there are a few methods available to extract plasmid DNA, including anti-exchange resins and membranes, and advanced silica membranes. The amount of culture used in these methods range from 1 to 5 mils. The culture media used impacts the growth of the bacteria. Increasing normal growth in Loria broth or LB broth can be increased to 2 to 4 times normal when using enriched medias like terrific broth. The temperature of the bacterial culture that it is grown at can also speed up or slow down this bacterial growth, which will affect the amount of plasmid in each of the cells. And then how long the culture is incubated at this temperature has a huge impact on the growth of the bacteria. So what are the different plasmid DNA purification scales? I mentioned it earlier. They're from 1 to 5 mils all the way up to 2.5 liters, which is a gigaprep. There are also commercial scale-ups of culture, which are usually done in a bioreactor. Today, we will be talking about the mini prep, which is again 1 to 5 mils, and maxi prep scales, which is 100 to 500 mils of culture. So plasmid DNA purity is also an important part of any extraction method. Endotoxin contamination is the most common concern for downstream applications using plasmid DNA. Therefore, most plasmid purification products have reagents to reduce endotoxin levels. So what are endotoxins? They are heat-stable lipopolysaccharide molecules associated with the outer membrane of certain gram-negative bacteria like E. coli. They are released by bacteria as they grow and divide in their normal life cycle, and large volumes are released during cell lysis. Endotoxin influences cell growth and differentiation and subsequently reduces transfection efficiency and protein expression levels in some sensitive mammalian cells. So plasmid purification methods have different purity grades, as shown at the bottom of the slide, depending on their final endotoxin levels. So the molecular grade or standard has greater than 10 EU per microgram of pDNA. Transfection grade or low endotoxin has a range of 0.1 to 1 EU per microgram, and advanced or endotoxin free has less than 0.1 EU per microgram. So challenges within manual methods for plasmid purification. First, they are very time consuming and with multiple manual steps. They are inconsistent with variation seen between users with, again, it's hard to get humans to do things consistently. Inconvenient, centrifugation, multiple touch points, and a lot of repetitive motion. There are quality issues between the various methods around purity, RNA and genomic DNA contamination, and supercoiled percentage. They lack scalability because workflows are dependent on extraction format and chemistry. And lastly, there's a large number of resources required to perform these extractions. So let's talk about the solution that we have here for a flexible automated mini scale plasmid isolations. So the MagMAX Pro HT no spin plasmid mini prep kit was developed with the following features in mind. It is a centrifugation free mini prep plasmid purification. It's the only kit on the market that is completely centrifugation free.
It utilizes a dual paramagnetic bead-based separation, which results in yields of up to 20 micrograms of DNA, plasmid DNA per mil of bacterial culture. It is transfection grade, and it is high throughput. High throughput because it was designed to be used on the KingFisher Flex and Apex instruments. It is also automation ready because it can be used on a TCAN Fluent or Hamilton Microlab Star with the integration of the KingFisher Presto instrument. Total processing time is approximately 35 minutes for 96 samples, with about 10 minutes of hands-on upfront preparation. The workflow involved in this kit is based on a novel 2-bead system where the backbind bead is added to the culture sample. This bead is highly positively charged to bind the E. coli, making it a centrifugation-free process. Once bound to the beads, the cells are lysed, which releases the plasmid DNA. Neutralizing the sample precipitates out all the cellular debris and genomic DNA, which is then removed, with the first bead leaving behind a cleared lysate sample. The PlasmidPro bead is then added to the sample. This bead is positively charged at the pH 5, which is what the lysate pH is at, which allows binding of the negatively charged plasmid DNA. Following 2 successive washes, the DNA is eluted off the beads when they are placed in a pH buffer of around 8 to 8.5, where the beads now have a more neutral charge, allowing the pDNA to be eluted off. So here's a general schematic of the workflow for the kit on a KingFisher instrument. So initially, the user must set up 7 plates, which will be loaded onto the instrument, 5 reagent plates, and 2 tip cone plates. Culture samples are added or grown up in the 96 deep well plate. Then the backbind beads are added with a quick mix before loading the plates and starting the script. After approximately 9 minutes, the instrument will pause, allowing the user to add a precipitation buffer, and then the run is resumed. About 6 minutes later, the instrument will pause a second time and allow the user to add endotoxin removal buffer and the plasmid pure beads. After restarting, it runs for another 15 minutes, and then you're able to collect your samples. So the total runtime is approximately 32 minutes. So here I've included some data generated using the workflow on both the KingFisher Flex and Apex instruments. The graph on the right shows representative data. The yields, again, are shown in blue. The purity ratios of 260 through 230, 260 to 80 in red in the middle, and then 260, 230 in green at the bottom are also shown. The data on the left side of the graph is generated using the KingFisher Flex with a large plasmid, approximately 11.4 kb, and then the data on the right is with a smaller 5 kb reporter plasmid. Both were transformed into top 10 bacterial cells and grown in LB media. The yields were consistent at approximately 17 micrograms on average, with purity ratios at the expected 1.8 and 2.0 values respectively. The figure on the left shows a 1% agarose gel loaded with 1 microgram of plasmid DNA, showing a majority of that plasmid DNA is supercoiled with no observable RNA or genomic DNA contamination. We also tested these samples for endotoxin levels and found that all samples tested fell within the 0.1 to 1 EU per microgram DNA range for transfection grade in the table on the left. The graph on the right shows transfection data generated using the 5.1 kb secreted alkaline phosphatase reporter plasmid. Transfection efficiency is on the y-axis and show that the samples generated with our workflow outperform those generated using manual filter-based workflows. This is likely due to the amount of supercoiled DNA seen in our workflow. Lastly, we sequenced samples generated with our workflow and compared quality and continuous read lengths and found that sequencing quality is better with the linearized plasmid, which is shown on top, the quality, while the continuous read length is similar between the two samples, with linearized plasmid producing long continuous reads with lower amounts of plasmid, which is shown in the graph on the bottom. So what are some of the advantages of automated mini prep plasmid purification? You can achieve transfection grade plasmid DNA. This is the only kit on the market that is totally centrifugation free. It is semi-automated with a dual paramagnetic bead technology. It is scalable, semi to fully automated, increased sample consistency with significant time savings, hours to minutes. And one of the best selling points about it is that it's REACH compliant and a very eco-friendly with no hazardous reagents. Now I would like to turn it over to my colleague Marie Gonzalez, who will talk about our new Maxi Prep fully automated solution.
Thank you, Ron. I'm Marie Gonzalez, an R&D scientist focused on nucleic acid workflows for our sample prep customers. I'll be talking about the fully automated scale plasmid purification platform that we have available. The KingFisher PlasmidPro Maxi processor and endotoxin-free cartridge is the only commercially available all-in-one walk-away solution for Maxi scale purification of plasmid DNA from overnight bacteria culture. Depending on a lab's capabilities, a traditional manual workflow can take anywhere between 90 minutes to 4 hours, and that is contingent upon a user's experience level. All-in-one here means the user is only required to supply the overnight culture, load the cartridge into the instrument, enter the sample ID, and press start. In less than 75 minutes, all that's required is for the user to remove the product tube from the cartridge and properly dispose of it according to the local regulations. Manual prep experience is not the limiting factor in this system. Everything is supplied, including reagents. This system eliminates the need for additional expensive equipment such as centrifuges, a vacuum manifold, or pipettes. The system is optimized to process 150 milliliters of overnight bacteria culture from an OD 600 range between 1 and 6 with LB media. And that is for construct sizes between 3 and 10 kb. So next I'm going to talk about some of the highlights of the performance of the KingFisher PlasmidPro system. We tested different construct sizes, and in the graph on the left is the yield, while the graphs on the right indicates the A260-280 and A260-230 ratios. The data is grouped by construct size from 2.7 kb up to 11.4 kb, where N is the number of purified samples represented in the graph. The yield and purity results indicate that the plasmid DNA is suitable for most downstream applications. The important thing to note here is that yield may vary on the plasmid copy number, size, culture growth conditions as well. So you really need to take that into consideration when you're trying to maximize your output. A select number of those samples were assessed for supercoiled isoform of the product. Greater than 80% supercoiled plasmid was produced on average when isolated with the KingFisher PlasmidPro system, as confirmed by agarose gel analysis. Those samples are compared to a set prepared by an experienced user with the PureLink XP Maxi kit. All samples were mass normalized in this gel image. The supercoiled content was confirmed with 1% agarose gel that ran at constant 75 voltage for 75 minutes. The gel was then stained with ethidium bromide and imaged with the iBright imager. And then densitometry analysis utilized the iBright analysis software. On the right, we see the supercoiled results grouped by construct size, which are consistent. And keep in mind, supercoiled DNA is the desired isoform for most downstream applications, and the automated samples have little to no genomic or RNA carryover. Applications such as transfection requires consideration as to whether the purified plasmid DNA sample also has endotoxin carryover. Previously, we showed that endotoxin-free is defined as less than 0.1 endotoxin units per microgram of pDNA. You can see for all the samples tested from the PlasmidPro system, that expectation is exceeded. A subset of those samples were assessed for transfection efficiency of the two plasmid types shown here, CAP or luciferase expression. The sample expression efficiency is compared relative to the efficiency of the plasmid DNA extracted from the manual kit, and the results are consistently equal or better. For the next few slides, we will provide case studies for a couple of the most common downstream applications for Maxi scale prep purification. Using the PlasmidPro system, we evaluated the performance of the purified plasmid DNA in a protein production workflow. In this instance, we transfected purified plasmid DNA using the Gibco XB293 expression system to look at antibody production. The following slide compares the use of purified plasmid DNA from the automated system against samples extracted with traditional column-based manual methods. This case study highlights the total yield of plasmid DNA for both light and heavy chain plasmids for human IgG antibodies isolated between the two plasmid purification methods. The graph on the left shows total plasmid DNA yield on the y-axis for both heavy and light chain sequence encoding plasmids. The navy is the automated purification, while the sky blue bars show the manual plasmid purification method. Following transfection utilizing the Gibco 293 expression system, we assessed the total protein yield. In this case, it was the human IgG antibody. The figure to the right shows the total protein yield in milligrams per liter for various operators, which demonstrates comparable yields with either the automated and manual workflows. We also evaluated the performance of purified plasmid DNA from an automated system for RNA synthesis. This workflow outlines the key steps for producing RNA from a target gene construct using plasmid purification methods and solid phase in vitro transcription, followed by RNA purification. You begin by engineering a construct with the gene of interest for RNA production. Next, you transform and grow bacteria culture with your construct, then purify the plasmid with your choice of purification method. A clean template is necessary for transcription. In this case, we looked at plasmid DNA purified with the automated PlasmidPro system and compared it to plasmid DNA from a column-based method. You then take the plasmid DNA for solid phase IVT. This involves immobilizing the DNA template on a solid support for efficient transcription, which in this case utilizes dynabeads, streptavidin for in vitro transcription. Purification of RNA from the IVT reaction is crucial to remove enzymes, unincorporated nucleotides, and other contaminants, and we show efficient RNA purification using the dynabeads carboxylic acid for RNA purification and the dynabeads RNA binding buffer. We have also looked at a separate study not discussed here today that Ron presented earlier. For no-spin plasmid mini prep kit for solid phase IVT, With No Spin Kit, we have demonstrated feasibility for early research customers who would be screening multiple constructs for RNA synthesis using the same workflow. For this case study, we demonstrated the feasibility of an automated workflow solution for in vitro transcription, starting with Maxi scale plasmid purification using the PlasmidPro system. Here we take a 4 kb plasmid that has the RNA sequence for IVT, transform it in top 10 bacterial strain, pick a colony and grow overnight culture for plasmid purification. Again, the Maxi scale automated plasmid purification and manual processes are compared. The graph on the bottom left shows the total plasmid DNA yield in micrograms on the y-axis for both automated and manual processes. An equal amount of pDNA from each of the 3 different methods was linearized, and solid phase IVT was performed using Dynabead streptavidin for in vitro transcription. In this case, we also compared the purified plasmid DNA from an alternate supplier, which was at a gigaprep scale, shown here in the green bars. The IVT yield from the 3 purification methods was comparable, indicated by the middle graph. The y-axis shows the RNA yield in the IVT reaction from plasmids purified with the 3 different methods. The tape station adjuvant traces on the bottom middle shows the quality of RNA in a crude IVT mix, starting from plasmid with each of the 3 methods. We then purified the RNA using the Dynabead's carboxylic acid for RNA purification and Dynabead's RNA binding buffer. This resulted in a similar amount of purified RNA shown here on the graph on the top right. There were also no differences in purity ratios for the purified RNA, and the tape station adjuvant traces shown here at the bottom right shows the quality of the purified RNA, which can be purified from the IVT mix. And with that, I open it up to the Q&A. Thank you again for allowing us the opportunity to present our automated plasmid purification solutions for the mini prep scale with the no-spin magnetic bead system and the Maxi scale option with the PlasmidPro Maxi processors.
All right. Thank you very much both for your informative presentations. We'll now start into the Q&A portion. So if you haven't submitted a question and you would like to, please do so now, and we'll try to get to as many as we have time for. So for the first question, Marie, can you swap out or change any of the reagents in the cartridge for PlasmidPro instrument?
Thank you for that. No, the system is designed to give you all of the reagents you need for plasmid purification. We do not recommend swapping anything out. It's not possible, because then that renders the cartridge not usable.
All right, thank you. Given the focus of the webinar, beyond just throughput, how does automation impact the consistency and reproducibility of plasmid yields and quality compared to traditional manual methods?
That's a great question. So what we presented here, we showed that the quality of from the automated systems is comparable from a manual kit. Really, what you're saving here is time from the manual prep. And as I mentioned before, the user experience is something that no longer has to be in consideration. You don't have to require a user have so much experience, for example, in order to ensure that you're going to get the maximum potential yield out of the system. And it definitely eliminates user error. So that's another time savings realized, because you're not having to go back and check your steps everything is already accounted for with these systems.
All right, Ron, we've got one. Can I use any culture media in the mini prep kit?
So the KingFisher Apex supports LB and enriched media, while the KingFisher Flex and Presto are compatible with the LB media. However, if you're using enriched media on the Presto system, you can try diluting your culture media before using in the system to improve efficiency.
And Ron, again for you, does copy number have effect on the quality of the mini preparation?
Yeah, as I said in the presentation, a high copy number plasmids yield higher amounts of DNA and are ideal for most downstream applications. Low copy plasmids can be extracted, but yields may be lower and may not be sufficient for all transfection protocols. For low copy plasmids, optimizing culture conditions, increasing culture volume, or performing multiple elution steps may help improve yield.
All right, Marie, are there specific storage requirements for the cartridges with the Maxi plasmid instrument?
Room temperature, ambient temperature is all that is required to operate the instrument and to store the cartridges. We do not recommend storing the cartridges in cold temperatures, like 4 degrees or anything like that, solely because the components in the reagents will precipitate out. So keeping them at room temperature is recommended.
And another one here for you, Marie. Is it possible to reuse the Maxi plasmid cartridges multiple times?
Unfortunately not. The cartridges are single use only.
Ron, can I use cell pellets or pelleted biomass for mini preps?
Yes. However, the MagMAX Pro HT no spin kit chemistry has been optimized to use fresh, non-pelleted culture media.
And then I don't have a KingFisher instrument. Can I use the mini preps manually?
Yes, you can. We didn't develop it with that in mind, so we don't have a qualified script, but you can easily use the workflow process that we have for our original Cartridge kit, and we do have and I have prepared a version of that optimized for the manual prep, I guess, to do it manually for the KingFisher, this new no spin kit, so that we can have that available. We can supply that to customers if they really need to do it manually.
All right. Marie, what happens if an error occurs or the run gets canceled? Do you lose your culture?
That's a great question. It actually depends on when the error or when the run was canceled. So if you've loaded your cartridge into the instrument, but you haven't pressed start, you can cancel the run at that point and still be able to reuse the cartridge. However, if at any point after pressing start, the cartridge will be rendered not usable. So if there is an error, let's say, just as an example, let's say power was lost to the instrument and it occurred while the cartridge was being, while a purification was being performed on the instrument. Once the instrument has power restored back to it, the system will detect the presence of a cartridge and just flush everything out. And that is just due to the nature of the system losing power and making sure that there are no leaks or any kind of exposure from the reagents or culture. So yeah, it just depends on when that happens.
And then, Marie, another one for you. What is the shelf life of those cartridges, and does the system recognize if reagents expired?
The current shelf life is 9 months from date of manufacture, and yes, the instrument will detect that expiration date once the barcode is scanned. However, if you want to proceed with using the cartridge beyond the expiration, that is something you are able to, but you will have to acknowledge that you're proceeding with an expired cartridge.
Thank you. Let's see here. Ron, for you, using terrific broth versus LB often increases endotoxin levels, creating problems in downstream transfections. Does the magnet technology in MagMAX kit overcome this issue?
So this is a great question. We get this a lot about the differences between using Terrific and LB. As long as the amount of biomass, so we have OD specifications for the kit. So if you're using Terrific broth, somewhere between 2 and 4 is preferable. If you stay within that the OD range, our system is optimized and designed to handle that amount of biomass, so that we should, with the use of the amount of endotoxin reduction buffer we have, you should still get transfection grade samples.
All right, thank you. Marie, are there volume limitations with the cartridge for the Maxi prep solution?
So the input volume is 150 milliliters of overnight culture. If you do not have 150 milliliters, we recommend that you bring the culture up to that volume with the media that was used. And there is a volume indicator on the cartridge as well. So as long as you're right there meeting that line, you should be fine.
Ron, what are your recommendations to increase yield with the KingFisher and MagMAX solution?
So to maximize yield, you need to ensure proper mixing of the backbind beads. We have a quick mix up and down 5 times as soon as you add the beads to the culture. Consider adjusting the script parameters to heat the elution buffer to 37 degrees before or during elution. And to increase DNA concentration, you could reduce the elution volume to 50 microliters per well instead of the recommended 100 microliters. And then the last thing is you could perform 2 elution steps, which is an easy script fix or change that to do 100 microliters of the elution buffer to get approximately 10 to 20% more of the plasmid DNA.
All right, it looks like we got time for a couple more questions here. Marie, can I use my own elution buffer with the cartridge?
No, as I previously mentioned, you're not able to swap out any of the reagents. That includes the elution buffer. The cartridge does come with Tris EDTA buffer pH 8.
And looks like this will be our last question. Ron, this is for you. Is the MagMAX solution flexible? Can I change my reagent volumes like elution to maximize concentration?
I'm sorry, can you repeat it? I was thinking, I think I may have already answered that.
Is the MagMAX solution flexible? Can I change my reagent volumes like elution to maximize concentration?
Yeah. So again, like I said earlier, if you want to increase your concentration, you can attempt using lower concentration. You can also, we do have the capability to modify the amount of solution you use to try to handle the biomass if you are using a little bit more biomass than what we might recommend. So this is definitely a flexible and we think scalable solution.
And we did get try to squeeze one more in here. Marie, are there plans to produce a PlasmidPro device with a capacity for more than one prep at a time?
There are plans for improved product offerings, yes.
Okay. So I'd like to thank you both for your presentations and the Q&A here, for your time and your important research. We'd also like to thank LabRoots and our sponsor, Thermo Scientific, for underwriting today's webcast. And before we go, I'd like to thank the audience for joining us and all the questions. Any questions that we didn't have time for or that were submitted during the on-demand will be addressed via the contact information provided when you registered. This webcast will be able to be viewed on demand, and LabRoots will alert you via email when it's available. We do encourage you to share that email with your colleagues who might have missed today's live event. So until next time, thank you very much again, and goodbye.